Conclave 2021 is our effort to reach out to general public, students, and academicians through exchange of ideas. The broad theme of Conclave 2021 is India at 75. Today is our first day of this webinar series where three students will be presenting on their viewpoints on three different topics. Before we actually begin with today's session, I request our guiding force, our principal, Professor Brinda Borkar, to say a few words.
Good morning all and welcome to Conclave 2021. It is my proud privilege that our students are also delivering the webinars. And the initiative taken by IQAC is really to be appreciated. During the pandemic, we started webinars by the teachers first. That was successful. Then we conducted the international symposiums, national symposiums, inviting the various eminent personalities in the field of science and academics. And when we were successful in that, we said that our students should be also trained for delivering the webinars. And I was very happy that the students, even last year, have done a commendable job in this Conclave 2021. It was appreciated by many people outside because one advantage of webinar is the YouTube remains and the YouTube can be seen by the rest at different times. Today is the first day of Conclave 2021. We have got a very good response for Conclave this year, and I am definitely sure that it will be again a big success. We are celebrating 60 years of our establishment, college establishment, 60 years of Goa's liberation, and 75 years of independence of India. Yesterday, there was a very nice lecture by Dr. Monday, who is Director General of CSIR. I wish that all the students go through that YouTube and he will take you to the journey of 75 years post-independence in science and technology. And where do we stand? Which are the areas that we can conquer and how can India be the leader? I hope all our science students listen to him and march in that way. So, at present, again, I welcome all to Conclave 2021 and I wish you all the success in this endeavor. I appreciate again IQAC coordinator, Dr. Shilpa Samant, as well as the team Conclave 2021, different teachers of different departments who have been coaching the students for webinar. All the best and thanks all.
Thank you, ma'am. Today we have invited our own Mr. Vishnu Chari to chair today's session. Mr. Vishnu Chari is well known in Dempe College, but still I would like to give him a formal welcome and also like to introduce him to the students and to you all. Mr. Vishnu Chari is presently an assistant professor at the
Shilpa, you are on mute.
Today, we have invited our own Vishnu Chari to chair this session. Mr. Vishnu Chari is not new to Dempe College. Everyone knows him very well. But still, I would like to formally welcome him and give a small introduction to the participants. Mr. Vishnu Chari is presently an assistant professor at School of Chemical Sciences, Goa University. He is also pursuing doctoral research in the area of computational chemistry at KK Birla Institute of Technology and Science, Goa campus. He has keen interest in understanding the catalytic cycle of organoxylium compounds as glutathione peroxidase mimetics for reduction of oxidative stress. With a degree in fire technology as well, he has conducted programs on fire safety for students as well as faculty members. A warm welcome to you, Vishnu. Now we shall proceed with our presentation. I hand over to Irena. Miss Irena, to take over, please.
I would like to introduce the student presenters for today. Ms. Sonali Viraj Maekar from FY BSc Biotechnology, who will be enlightening us on the topic India at 75, Biotechnology Then and Now. Where our second speaker for today, Mr. Sherwin Vinay Suarez from SYBSc, who will be speaking on the topic Advancement in Biotechnology and how it aids in medical research in India. And our last speaker, Ms. Ramira Mascarenas from TYBSc, will be speaking on the topic particle beam technology and medical science. Everyone in the audience, kindly take note. The question and answer session will be taken up at the end of all the three sessions. I would request the first speaker for today, Ms. Sonali Maekar, to take over.
Well, a very good morning to everyone. I am Sonali Maekar. I'm a first year BSc Biotechnology student. Well, is my screen visible?
Yes, Sonali.
Well, India has developed progressively in the past 75 years, and we have seen some excellent growth. And biotechnology sector has definitely been one of the key factors in this. So today, under the guidance of my teacher, Ma'am Preeti, I would be talking about the situation pre-independence and post-independence and how the biotechnological sector has played a part in this. First up, let us start by knowing what exactly is biotechnology. Biotechnology is any technological application that uses biological systems, living organisms, and derivatives to make or modify products or processes for a specific use. Now, this technological application is used to manufacture medical and veterinary products, agricultural products, chemical products, biological products, and even in pollution control and waste management. During the recent 2-3 decades, biotechnology has brought a total revolution. We have seen advancements, we have seen sophistication, and a proper exploitation of all chemical, physical, and biological process. Now we have different sectors to biotechnology. Them being, we have medical biotechnology, we have industrial biotechnology, we have the environmental biotechnology, and we have marine biotechnology. Let us move on to discussing what was the situation of healthcare pre-independence. In the early 1900s, we had a very poorly developed healthcare infrastructure, not enough medical equipment, and a very poor doctor-to-patient ratio. Though we had Ayurveda and other regional and plant-based medications, we had less than 2,000 properly equipped hospitals across India. We had a widespread effect of various communicable diseases such as TB, malaria, smallpox, and this affected our huge population. We also had a very high infant mortality rate and a maternal mortality rate due to the lack of preventive care. We also had a lack of awareness among the people, and the flu pandemic of 1918 killed around 12 million Indians. The World War, and India's freedom movement also led to a lot of youth physically disabled. Now, what is the situation post-independence? India's healthcare system has evolved drastically. By the time of India's independence, we had around 7,500 hospitals. We have also eradicated smallpox and we have also eradicated smallpox and polio by the means of mass vaccination and proper medication. We have built our healthcare infrastructure and we have evolved methods for surgical procedure and diagnostic techniques. Now, how has biotechnology helped us achieve this? We have had production and export of vaccines. We have discovery and production of possible solutions for various diseases. We have had development of diagnostic techniques such as ELISA and PCR. Now, what are the biotechnological advances we are looking at? Well, we have genetic screening of diseases. An individual can scan his or his DNA code to look for unknown mutations linked to a disease. Newborns are often screened at the birth to identify treatable genetic disorders. For example, newborns are scanned for disease called as severe combined immunodeficiency. Now, individuals with this genetic disease lack a fully functional immune system and usually die within a year. However, due to regular screening, this newborns receive bone marrow transplant, which has a 90% success rate in SCID. A well-known example in adult screening is for mutations in the BRCA1 and BRCA2 gene as a risk factor for developing breast cancer and ovarian cancer. We also have recombinant protein production. Now, this technology allows scientists to introduce human gene into microbes to produce human proteins that can be introduced back to patients to carry out vital functions. In 1978, the company GeneTech developed a process to recombinantly produce human insulin, a protein needed to regulate blood glucose. Recombinant insulin is still used to treat diabetes. We also have a gene therapy. Now, the goal of gene therapy is to replace a missing or defective gene with a normal one and to correct the disorder. Now, gene therapy is being tried for sickle cell anemia and also for SCID. Now, the first gene therapy was given to a 4-year-old with ADA deficiency. We also have chimeric antigen receptor T cell therapy. Now, this is a technique to help your immune system to recognize and kill cancer cells called cancer cells. Now, T cells from a cancer patient are isolated and genetically engineered to express receptors that allow them to identify cancer cells. Now, when these modified T cells are put back into the patients, they can help to find the cancer cells and kill the cancer cells. This has shown promising results in end-stage cancer patients, particularly the ones suffering from lymphatic leukemia. Now, let us move on to the agricultural sector. Now, what was the situation of agriculture before independence? Well, the agricultural sector was very prone to crumbling and had a very and was very highly volatile. There was also no stable production rate for various crops. Also, the land used for crops were used for cash crops. There were crop failures and famines and mass starvation. We have the Bengal Famine of 1943, which killed a huge population of 3 million Indians approx due to starvation, malaria, and other disease aggravated by malnutrition. The economy was crashing and people were dying because of starvation or deficiencies caused due to malnourishment. Though in today's world we do not have people dying of starvation, we do have people dying of hidden hunger. So how is which is malnourishment? So how is biotechnology helping us tackle malnourishment? We have biofortification. Now, biofortification is genetically modifying crops to have an improved nutritional value, have an improved quality and content of proteins, oils, and vitamins. One example is hybrid maize, which has twice the amino acids lysine and tryptophan. We have Atlas 66, which is a wheat variant and it has a very high protein content. We also have carrots, pumpkins, and spinach, which are vitamin A enriched. Next up, we have single cell proteins. Single cell proteins are microbial proteins and they are edible unicellular microorganisms. The biomass of protein extract from pure or mixed cultures of algae, yeast, and bacteria are used as an ingredient or a substitute for protein in rich foods. Examples are spirulina. Now, how is biotechnology helping us tackle crop failure? Well, we cannot afford to lose crops for our huge population. But over the period of time, we have witnessed large-scale use of agro-based chemicals such as fertilizers and pesticides. But this come with their own set of drawbacks. They are very harmful for the environment and even to us on consumption. So through biotechnology, we have crops which have been genetically engineered to make a protein that kills certain insects when they feed on them. The protein from the soil bacterium Bacillus thuringiensis, which is the Bt toxin. We also have mutation breeding, where mutations are induced artificially to get a desired trait. Now, basically, this method is used to transfer disease-resistant gene to a very high-yielding variety. We have Pusa Sadabahar, which is a chili, and which is a chili variant and is resistant to chili mosaic virus. We also have Pusa Swarnam, which is a mustard variant and is resistant to white rust. We have the Green Revolution, which took place in India in the 1960s, and this was done by introducing high yield varieties of rice and wheat to increase the food production in order to alleviate hunger and poverty. We also have the White Revolution or the Operation Flood that took place. Now, this was a step taken by the Indian government to develop and help the dairy industry sustain itself economically. We have often come across the term transgenic animals. Now, transgenic animals are animals that have had a foreign gene deliberately inserted into their genome. Transgenic animals are used for various purposes, such as studying human diseases such as cystic fibrosis or cancer or Alzheimer's, or for the production of biological products such to study the contribution of genes in the development of disease, for vaccine safety, and for testing chemicals. Now, how has industrial biotechnology been helping India? Well, we have large-scale production of biofuels such as ethanol, which is being produced from corn and sugarcane, and biodiesel produced from algae and plant sources. We also have biogas, which is the goba gas. We have biopesticides, which have been derived from natural materials like animals, plants, and bacteria. We have organic acids such as acetic acid and citric acid. We also have production of dairy products such as various different types of cheese or yogurt or probiotics, and production of alcoholic and non-alcoholic products. Now, what is the future of biotechnology in India? Well, we are looking forward to cure of various lethal diseases that are not known as yet. We are looking forward to global safety and security. We have food security and personalized medication. Thank you for your time. If there are any questions, I would love to answer them.
Am I audible? Hello? Hello?
Yes, you're
Am I
audible. Yes.
Yes. So I have a question for you. So as we all know, we are currently in the pandemic era, and because of the virus in play, there are various factors which are affected negatively, and there's a lot of turmoil. So I want to know, how did biotechnology exactly help for the people who are in pain and who are suffering?
Well, during the COVID, we have had testing kits that were indigenously made. We also have vaccine development that took place in India, and we have had clinical trials, and we have successfully established something. We also have developed safe trucks, basically, and we have manufactured personal equipment, and we have had a large-scale production of oxygen also, and we have had disease surveillance done in India.
Thank you.
Participants, can we take the questions at the end of the session? The participants kindly take note that we'll take all the sessions, take all the questions at the completing of all the 3 sessions. Thank you, Ms. Sonali, for enlightening us on biotechnology then and now. I would request our next presenter, Mr. Sherwin Suarez, to take over.
Mr. Sharma, you are not audible.
Am I audible now?
Yes.
Alright. So, my name is Sherwin Suarez, and I will be speaking on the advancement in biotechnology and how it aids medical research in India. So, the word biotechnology has received enormous importance and significance during the 2 decades, which is just unprecedented. So far, biotechnology has touched our lives in all aspects, such as food, health, and animal life. We have also noticed the importance and potential of biotech for the improvement of our environment and for better living, as well as India is among the top 12 destinations for biotechnology worldwide. The industry comprises 2,700 biotech startups and 2,500 biotech companies in the country. India has 44% of the global abbreviated new drug applications and 1,400 manufacturing plants which are compliant with WHO regulations. History of medicine in India. The Chronicle of Medicine is the story of man's struggle against illness as early as 5000 BC. India developed a comprehensive form of healing called Ayurveda. One second, there's a small issue. Sorry about that. There was a small issue with the presentation. I'm going to continue. So, history of medicine in India. The Chronicle of Medicine is the story of man's struggle against illnesses as early as 5000 BC. India developed a comprehensive form of healing called Ayurveda. Ayurvedic treatments have been documented. Are my slides visible?
The slides are not changing. You may sort of press Control and Alt and means while presenting and select that window only where there is a full screen.
One second, there's a small network issue. All right. So, sorry about that. Network issue. All right, there was that, yes. Ayurvedic treatments have been documented in books for many generations. The knowledge was also orally transmitted, but mostly written in palm leaf manuscripts. There are two treatments in Ayurveda, purification therapy and elevating palliative therapy. Although cosmetic surgery is a modern development, the origins date back more than 2,500 years during the 6th century BC. An Indian sage known as Sushruta, the father of surgery, documented more than 1,100 diseases, catalogued the use of various medicinal herbs, and wrote directives for performing surgical procedures, including rhinoplasty and skin grafts. Medicine has changed considerably over the centuries. The technology today provides us with 3D printed body parts, MRI displays, malicious tumors, and medical remedies are produced in laboratories. Although none of this existed in ancient India, medics to manage to treat patients using a natural system, such as Ayurveda. Sushruta, as I mentioned earlier, procured many operations and recorded them in his Sumita, paving the way to modern medicines and science in India. India, a biotech growth catalyst. For the last 20 years, biotechnology is one of the fastest emerging branches in India. Demand of biotechnology professionals is increasing every year in India as lots of biotechnology companies are seeking India as a potential market. It's among the top 12 destinations for biotech in the world with approximately 3% share of the global biotech industry. As I mentioned earlier, India is the largest biotech destination in the Asia Pacific region, with over 4,237 plus biotech startups expected to reach 10,000 by 2025. 760 plus core biotech companies, 200 plus biotech products, and rising contribution to national GDP, 2.7% in 2020 against 2.2% in 2019, which was a whole lot. What are the origins of biotech in India? So the origins actually of biotechnology, the term was originally coined by the Hungarian scientist Carol in 1990. The biotechnology industry in India can be traced back to the 1980s when the government created the Department of Biotechnology India. From merely a 1.1 billion industry in 2003, it has grown exponentially in size to a $51 billion industry as of 2018. Seeing India's humble but natural progression in medicine, it should come as little surprise that India is one of the fastest rising forces of biotechnology in the world. Some advantages of biotechnology in India are its skilled human capital. With its large population, there are an ample amount of doctors, engineers, medical staff, and people who can work these technologies in innovative ways. And with the rising youth population as well, there will be more innovative ideas and more uh, value added to this skilled human capital in the coming years. Infrastructure facilities, as we have gone through the pandemic, we other governments around the world have realized the importance of biotech in society. As a result of which, many governments, including ours, had contracted many biotech firms to establish better infrastructure facilities for these biotech firms. Policy support, as I mentioned earlier, governments have contracted these biotech firms for increasing supply of vaccine, epidemiological factors as well. So, some wonders of medical biotechnology in medicine. Medical biotechnology has improved tremendously in the recent decades, which led to the multiple innovative techniques that aim to prevent, diagnose, and treat diseases. Some of these are stem cell treatments. Stem cell treatments you can use for regenerative medicine, tissue engineering, and antibiotics. Targeted cancer therapies. These type of cancer treatments that targets proteins that control how cancer cells grow, divide, and spread, and you can target these specific proteins. CRISPR, 3D printed organs, which would play a huge role in treating a lot of people, as there are many people who are put on organ waiting lists, and because of that, they don't get to experience life. Their lifespan is cut short, unfortunately, and it also has the potential to increase expectancy, health, and quality of life in general. Some of the top biotech breakthroughs are spit tests for cancer, prosthetic feedbacks, as I mentioned earlier. You can also, with the help of biotechnological techniques, you can create artificial prosthetics for people who have lost their limbs as a result of some accident. So these limbs you can control via nerve signals, nerve impulses, and you have rapid deployment of vaccines. As technology improves and becomes more efficient, we can also increase our rate of production vaccines and can help prepare us as a society for any later uh, epidemics or pandemics in the future, which could pose a huge threat to our civilization as a whole. Then you have bioplastics and bio energy as well, both of which are, as I said, a very clean natural resource and can be biodegraded. And another bioenergy is also a clean form of energy that produces little to no waste. So, what does the future of biotechnology therapies look like? So, as I mentioned earlier, stem cells can be used for various purposes, including the regeneration of tissues, grafting for skin, as well as nerve regeneration. And you have nanomedicine research as well as new drug delivery systems that you can use to deliver drugs to specific parts in your body, specific points in your body. And you have biotechnology, which is also revolutionizing the diagnosis of diseases caused by genetic factors, some of these factors which are unavoidable. So these new tests can detect changes in the DNA sequence of genes associated with disease risk and can predict the likelihood that a person or patient will develop a disease and can treat it. Though there are some medical and ethical issues of biotechnology, which I have listed, some of these are risk to human life in clinical trials. So, as our society progresses and becomes more progressive, many people look down upon the harsh treatment of animals in clinical research. You have animals, rats, rabbits being used in trials, and many of these animals are kept in very poor conditions. As a result of this, many awareness groups have been protesting the use of these animals, as well as humans who voluntarily participate for these clinical trials. Though sometimes with human clinical trials, some of these drugs are untested, so these can lead to medical problems and can also, unfortunately, sometimes lead to a loss of life. So another point is high cost may exclude the poor. As I mentioned, biotechnology is an emerging field, though it is quite prevalent. It is also still emerging. So the technology being used is quite expensive, and the techniques and systems are also quite expensive. Let's take, for example, stem cell treatment. Not many people will be able to afford regular stem cell treatment. So this kind of treatment in the future may be subsidized, but for the time being is expensive for the majority of people. Privacy concerns. As I mentioned in the previous slide, there are biotechnological techniques that can help study the human genome and the whole DNA sequence as a whole, allowing us to predict a person's ancestry, be able to tell a person's ancestry, predict if they will develop a certain disease later on in the future, or if something is recessive, something is dominant. It can help to basically unravel the mystery of a person's full body. And because of which, this can pose a lot of privacy concerns, as people see this as an invasion of privacy, even though it can help this person avoid certain life-threatening diseases later on in the future, which they are not aware to. And some groups also oppose stem cell research, as the procurement of stem cells from blastocysts in humans can also damage these blastocysts, which can lead to an ethical dilemma, as should we extract stem cells, and if so, what are the uh, boundaries that we are willing to cross with this? So in conclusion, Indian biotechnology is built on entrepreneurship, innovation, developing domestic talent, and demonstrating value-based care. Given the long history of diseases in India, the country has accumulated years of experience and scientific knowledge to prevent and treat them. India is working to boost the biotechnology sector under the flagship programs such as Make in India and Startup India. And given India's fast growth ever since our independence, we are already neck and neck with most Western countries. My references, and thank you very much. I'll hand it over to ma'am.
Thank you, Mr. Sherwin, for the insights on advancement in biotechnology and how it aids medical research in India. I would like to request our last presenter for the day, Ms. Ramira Mascarenas, to take over.
Good morning, everyone. My name is Ramira Mascarenas, and under the guidance of my professor, Dr. Mishkil Naik, I'm presenting on particle beam technology and medical science. In this session, I will briefly explain the history and development of particle physics in various fields, the applications of particle physics in medicine, and will also discuss the future advances of particle physics in medical technology. Our story starts shortly after the moment of creation. Cosmologists hypothesized that all matter in the universe was contracted into an infinitely small space called a singularity and then exploded, producing an enormous fireball at a temperature of 100 billion Kelvin. During the first milliseconds of this cosmic fireball, the universe was filled by a great density of energy in the form of radiation. The energy state was continually altering from electromagnetic radiation to electrons, positrons, and back again to radiation. A positron is an anti-electron, that is, an electron with a positive charge. Modern particle physics research is focused on subatomic particles, including atomic constituents such as electrons, protons, and neutrons, that are produced by radioactive and scattering processes. Such particles are photons, neutrinos, and muons, as well as a wide range of exotic particles. Particle physics makes a difference in your life. From the phone you're holding in your hand right now to far away in space, particle physics finds its applications. Did you know that the World Wide Web is only possible because of particle physics? Or that medical accelerators have treated more than 30 million people around the world? Is there really a limit to what particle physics can do? There are more than 30,000 particle accelerators in operation around the world today, and they shrink tumors, make better tires, spot suspicious cargo, clean up dirty drinking water, help design drugs, discover the building blocks of matter, and do so much more. We'll be discussing a few common but important medical applications of particle physics. Isotopes in medicine. The first uses of isotopes in medicines were in radiopharmaceuticals, and this is still the most common use. Deuterium in deuterated drugs, carbon-13 used in liver functions and metabolic tests are some examples of non-radioactive medical isotopes. Technetium-99m and iodine-131 are examples of radioactive medical isotopes. In today's world, doctors order X-rays to diagnose all sorts of problems, from a broken bone, pneumonia, heart failure, and much, much more. We barely think about it, but not so long ago, a broken bone, a tumor, or a swallowed object could not be found without cutting a person open. Wilhelm Röntgen, a German professor of physics, discovered X-rays in 1895, accidentally, while testing whether cathode rays could pass through glass. Because he did not know what the rays were called, he called them X, meaning unknown rays. Röntgen quickly found that X-rays would pass through human tissue too, rendering the bones and tissue beneath visible. For his work, he received the first Nobel Prize in Physics in 1901. X-rays are commonly produced in X-ray tubes by accelerating electrons through a potential difference and directing them onto a target material. The incoming electrons release X-rays as they slow down in the target. The Crookes tube was the first generation X-ray tube, which was improved in 1913 to the Coolidge tube. Excuse me. There's a technical error. Yes. Okay. It's fine, sir. Sorry. Similar to light, X-rays are part of the electromagnetic spectrum with a wavelength in the range of 0.01 to 10 nanometer and energies in the range of 100 electron volts to 100,000 electron volts. Unlike light, however, X-rays have higher energy and can pass through most objects. Medical X-rays are used to generate images of tissues and structures inside the body. In the picture, you see some uses of X-rays and their corresponding energies and wavelengths. X-rays traveling through the body pass through an X-ray detector on the other side of the patient. Here are a few X-ray scans you're probably familiar with. The discovery paved the way for the development of today's broad spectrum of imaging techniques, including magnetic resonance imaging, MRI, computed tomography, CT, ultrasound, echocardiography, and many others. Advances in X-rays. Using groundbreaking technology, researchers are testing a new method of X-ray imaging that uses color to identify microfractures in bones. Microfractures were previously impossible to see using standard X-ray imaging. Another group of researchers have worked on making X-rays much more flexible. New technology means that X-ray detectors could one day effectively be printed onto any suitable surface. So they could be made curved to wrap around the part of the patient's body being scanned, produce a much more accurate image. Now let us look at another technology commonly known as the CT scan. In 1967, Sir Godfrey Hounsfield invented the first CT scanner at EMI Central Research Laboratories using X-ray technology. In 1971, the first patient brain CT was performed in Wimbledon, England, but it was not publicized until a year later. He won the Nobel Prize for Medicine in 1979. In this slide, you can see the inside working of a CT scanner. CT refers to a computerized X-ray imaging procedure in which a narrow beam of X-rays is aimed at a patient and quickly rotated around the body, producing signals that are processed by the machine's computer to generate cross-sectional images or slices of the body. These slices are called tomographic images and contain more detailed information than conventional X-rays. Once a number of successive slices are collected by the machine's computer, they can be digitally stacked together to form a three-dimensional image of a patient that allows for easier identification and location of basic structures, as well as possible tumors or abnormalities. Instead of a film, CT scanners use special digital X-ray detectors which are located directly opposite the X-ray source. On this slide, we see an image of a CT scanner, and to the right are two abdominal CT scans. The yellow circle indicates a detected tumor. Studying ancient diseases and wounds is important as we can study the cancer of the past, and this can be useful for modern medical research. Now we'll see MRIs. Dr. Raymond Damadian was among the first to propose the use of nuclear magnetic resonance to produce medical images. He hypothesized that cancerous cells could be differentiated from non-cancerous ones using magnetic resonance. He theorized that cancerous cells hold more water and would show up in MR due to the increased number of hydrogen atoms in relation to the extra water. Damadian then performed an experiment with both cancerous and normal rats, which proved his theory. The image on the right shows the patent of this technology. MRIs employ powerful magnets, which produce a strong magnetic field that forces protons of the body to align with that field. When a radio frequency current is then pulsed to the patient, the protons are stimulated and spin out of equilibrium, straining against the pull of the magnetic field. When the radio frequency field is turned off, the MRI sensors are able to detect the energy released as the protons realign with the magnetic field. MRI scans are significantly more detailed than those produced by X-rays and CAT scanners. In the next slide, I'll play a video which shows an MRI acting on a metal stapler. Anyone who has had an MRI knows that MRIs fling metallic objects away due to their strong magnetic fields. Also, for humans, an MRI is completely painless, and there are no moving parts like in a CT scanner. Advances in MRI. Claustrophobic people are usually given mild sedatives to help them relax before an MRI scan. However, some clinics operate this newly designed MRI scanner. The magnetic field of the Earth at the equator is 0.00003 Tesla. Most MRI scanners operate at 1.5 Tesla. An immensely powerful 10.5 Tesla magnet weighing almost 3 times more than an aeroplane is the most powerful MRI used on a human being yet. And still, the most powerful MRI in the world operates at an incredible strength of 11.7 Tesla. A lot of research is being conducted to make these technologies large enough to scan an entire truck in one go, and also small enough to be portable to quickly carry to a crowd to inspect suspicious cargo. Thank you. If you have any questions for me, I'll be glad to answer.
Thank you, Ms. Ramira, for providing us a great insight on the topic particle beam technology and medical science. I would like to request our participants to put forth your questions in the chat box for the question and answer session. Handing over to Ma'am Shilpa.
Yes, anyone wants to ask any question, you can either ask or you can put it in the chat box. There is a feedback link which has gone uh, 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 into the chat box. Please fill that. The certificates will be given based on the feedback which is given by the participants. We require the feedback in order to improvise it in the later for the next sessions of Conclave. Thank you.
I have some questions for the participants. Can I ask?
Sorry, Mr. Vishnu, did you say anything?
Yeah, I have few questions to the participants. Can I ask?
Yeah, you can ask, definitely.
Okay. I have one question to Ms. Sonali. So, what do you think will be the effect of introduction of or animals on biodiversity?
Sorry, sir, could you please repeat that?
Yeah, what do you think will be the effect of introduction of genetically modified crops or transgenic animals? Suppose we try to introduce them in the biodiversity, means rather than doing this thing in restricted area, suppose we allow this thing to happen in wide scale. What will be the effects there?
We might not exactly know if they can reproduce. So if they can reproduce, we have new species we can save, or
Okay,
we can actually diversify our ecosystem.
fine. Thank you. Uh, Mr. Shavin, I had one question for you. Yes, it was actually both means I had liked all your presentations. Everyone has presented very nicely, and all the talks were quite engaging. Mr. Shabin, what do you on in COVID manufacture? Am I audible?
One second, could you just repeat that?
Yes, Vishnu, you can continue. You are audible.
Okay, yeah, thank you, ma'am. So, Mr. Shabin, what are your views on what is the effect of indigenous COVID vaccine manufacture on current state that we are seeing right now, like in terms of medicine that we see with respect to COVID treatment?
I'm so sorry. Could you repeat the last part of what you said?
Uh, yes, we have indigenously manufactured COVID vaccines in our country. So, what do you
Yes.
think? What will be the effect of, or what are the effects that we are seeing on the current state for the treatment of COVID in India?
Well, for starters, as you said, yes, we have indigenously uh, manufactured a lot of vaccines in India, so much so that other countries have contacted us in response to ask for some of these vaccines, some of the treatments, and because of which it has helped a lot of people and has saved a lot of lives. We can also look at it as a form of another form of boosting the economy as well, as because of this large production of vaccines, India had taken one of the top spots in countering the COVID epidemic that we have been suffering from for the past almost 2 years now. Did that answer your question?
Yeah, yeah. Nicely you have answered. Thank you.
Thank you.
Hello,
Yes.
can I ask a question? Hello,
Yes, sure. Go ahead.
uh, I would like to ask a question to Ramira Mascarenes regarding that CT scan and MRI. Dear Ramira, most of the time when hello, is Ramira online?
Sorry, sir, could you please repeat?
Yeah, I would like to ask a question to you. During the whenever the CT scan and MRI is done, before that they give an injection. Okay, why they gives an injection and keep some time and then they conduct MRI or CT scan. What is the purpose of that injection?
Yeah, so sir, those injections are dyes and they help make the MRIs or CT scans appear clearer or more. It gives a better image, better quality to
Okay,
the image.
okay, but what is the this thing effect for of that injection? Okay, but what kind of injection? It is like a chemical or what is or
Yes,
this injection? It is made up of what like?
sir.
uh, these injections are made up of it's like a what to say whether it is like a vaccine like or just like a normal injection but
Yes, so the injection contains elements that can be detected in the MRI or CT scan. Sometimes
okay
some people have allergic reactions to these dyes. So
okay
before being injected, you have to discuss it with the radiologist
okay okay
to minimize any risk.
okay or what kind of risk it's like what kind of radioactivity risk or what
No,
kind
no,
of
no,
risk
no, just an allergic reaction.
okay allergic reaction of what radiation you mean to say
Not radiation, just the dye. There's no radiation risk in an MRI. However,
okay
in a CT scan, there is a radiation risk, though it's very, very small.
okay
Yes,
yeah thank you.
yes, you're welcome.
There are certain questions in the chat boxes. Students, if you can take them.
Yes, ma'am. Candice, you have asked me about bioplastics. So yes, bioplastics, unlike regular plastic, is biodegradable, which is one of the biggest problems we tackle with plastics. It has reached a point where there is literally a giant patch of land made up of trash floating somewhere in the Pacific Ocean. So, in this regard, bioplastics, which is biodegradable, would play a huge role in tackling environmental problems and help planet, keeping our planet clean and well, prolonging our species as a whole. That answer your question, Candice?
Yeah, Sherwin, thank you so much.
Right.
There are few more questions in the chat box. Students, can you please take it? They have taken the name, Sonali.
Well, Sumuk, your question was
There are
regarding
questions for you too.
Yes, ma'am.
I have questions for you.
Yes,
Can you
ma'am.
please see them
Yes.
in the chat box and answer?
Well, Sumuk, your
Even
question
their
is
mentors
about
can.
yes. Yes. Well, we do have genetically modified organisms, but there are laws put in place that actually make sure that these organisms are not wrongly used. And also, we have testing and research done and trials done before they are put out to the public. So, yes, these are safe and they can replace the predecessors. And also, why wouldn't we want to consume, suppose, grains and crops that have been enriched with a lot more nutrients than what we would get regularly? I hope that answers your question, Sumuk.
Thank you.
Um, um, Hello. I'll answer the question by Sangavi, how MRI produces images. So an MRI basically uses and sends magnetic and radio frequency into a patient's body, and the energy emitted by the atoms in the magnetic field sends a signal to a computer. So then the computer uses some mathematical formulas to convert the signal to an image. Yeah, hope that answered your query.
Okay, if the question answered, then can I request Mr. Vishnu Chari to give his comments?
Yes, thank you, ma'am. It's always a pleasure to come to my college, and I congratulate the students as well as their faculty mentors for this well thought out research presentations. Also, entire team who has in making this a very successful event. Um, Overall, with respect to the presentations, all the presentations were very engaging and kind of has given very good insights into the topic what students have chosen. The first presenter, Ms. Sonali, has given a good overview of the status of biotechnology from pre-independence era to the current stage, and also she has given some insight into how this particular or how the advancement of biotechnology may be used to tackle some problems which can be there in the future. Then Mr. Sherwin also has given a good idea about what kind of conditions of medical treatments that used to be there in India pre-independence, and how biotechnology have revolutionized the kind of this particular field to kind of detect means from diagnostic to the therapeutic aspect of these particular conditions. He has also kind of talk about what could be the challenges as well as the limitation faced by the state of the biotechnology development, and overall the presentation was quite good. The the last speaker, Ms. Ramira, has given a good idea, or basically she has kind of talked about lot of non-invasive methods which are currently used in medicine, and thanks to the particle beam physics or particle beam technologies that let us kind of explore these areas, and how the particle beam methods can be utilized for non-invasively identifying the cancer, specifically cancer, and how to kind of identify and diagnose this particular diseases have been nicely given. I thank Ma'am Shilpa and Principal Ma'am for giving me an opportunity to chair this session. And I once again congratulate all the participants as well as their mentors who have put in their efforts to make this event very successful. Thank you.
Thank you, Mr. Vishnu. Mr. Oro, Mr. Vishnu has answered your question in the chat box about the injunctions which you asked.
Oh, yes, ma'am. I got it. Thank you. I got it.
Okay, thank you.
Thank you very much. Yeah, yeah. It's a
Over to
wonderful session.
yeah, thank you. Ms. Irana, over to you.
Thank you, ma'am. Today's session cannot come to an end without thanking our principal, Professor Vrinda Bodkar, for always encouraging, motivating, and providing immense support for conducting innovative sessions like this. I would also like to extend my gratitude to the chairperson, Mr. Vishnu Chari, for giving us his valuable time. I want to congratulate the student presenters for today, Ms. Sonali, Mr. Sherwin, and Ms. Ramira, for widening our horizon on these topics. A big thank you to the mentors, Dr. Mishkil Naik, Dr. Preeti Pandit, and Ms. Alisha Malik, for guiding the students by being their mentor. And last and not the least, to all the students participants who have been there for today's session And thank you to one and all. We shall meet again with an interesting session on Conclave 2021 on Wednesday, 24th November 2021. Thank you and have a nice day. A gentle reminder to all, the participants kindly fill in the feedback form which is in the chat box. Thank you.